have us take a look at the webpage of Biomodels. It's hosted by the EBI in Kingston, and the Biomodels database is a fully proper curated database of biological model information. So that is structural information about algorithms that are supposed to represent biological knowledge in terms of an interaction structure. So whereas in the previous session, 2 weeks ago, if you attended our web support seminar, we have used MATLAB matrix functionality to set up networks matrices and display matrices. The end result was a static network. But in biology, what we see is that the networks that actually guide in vivo function, they are not static at all. And they do things according to the rules of interactions and to the strength of interactions between the nodes. But what we'll see today is that this interaction does not necessarily have trivial results in either way. So because we have also non-trivial results, the Biomodels database was generated to collect anything that people do trying to explain biological phenomena and have them available in a way that can be shared easily. So if you have followed the instructions, there is a search window in the database. And if you type in the model that you're looking for example, the glycolytic model developed by Nielsen, then it means that you will get a number of models associated with that name. And in our case, the idea was the glycolytic pathway model, which is in with a data bank ID, and by clicking on the data bank ID, you will be brought to the page that contains the model proper, but also information concerning the model. So if you click through the tabs, you will get overviews of the reactions that have been modeled, of some of the mathematical structure that is around it, and you can click on it to unfold the actual representation of the equation that guides the evaluation of the model. There are a couple of comments on the parameters and on the settings of the model. And finally, there is, of course, a history of the curation of this model, where it came from, in which version it is currently, and what types of things have been done to the original model and by whom. So on the left-hand side, somewhere where my cursor is currently located now, you have an option window where any of the versions that is available now can be downloaded. But as a start with, if there is a curated form of the model, this is the one that we want to choose for download. It's important to check this because there's also non-curated models and they were automatically generated. So if you have a network setup like the one that we did in the previous session, if you have a static network information about some set of reactions that form a functional module, say an anabolic module to synthesize a certain compound in a cell, then you can use this network information to automatically generate a model based on the assumption that it follows, for example, standard enzymatic kinetics to convert one metabolite into another. If that is the case, then you don't have a curated form, but if those are models that are actually documented with a proper paper publication and there's figures that have been published, then the curated form is in a way that the model in its default setting should be able to create or recreate one of the figures in an earlier publication. So if we click on it, you can see it already downloaded on my screen here. And if I show you the screen later, you'll be able to find the proper pathway. If you download it yourself in this moment, you need to go to the download folder set in your web browser. So if I go just to the welcome page, Biomodels database collects by now hundreds of curated models, and that is manually curated models, and a large repertoire of automatically generated models, which of course is not valuable as something where you want to already see the proper output, but which is something that you can use as a basis to develop a model of your own, where you would do the curation and the actual model development. The web page is maintained properly, and on the right-hand side you can see they feature some interesting introductions to the modeling process in a place called Model of the Month, where every month they pick one of the recently published models or recently implemented models and give a short description on what it does and how it works and what it is being used for. If you further browse the web page, you can see there's also something that is disease related. So there's a section that relates to models for specific diseases, and that is of particular importance for the biomedical community. And so depending on your interest, you might find many starting points to connect with biological research.